Mechanoreceptors and their stimulation mechanism. Sensory receptors. Sensory receptors are specialized to respond to changes in the environment or stimuli. Action results in graded potentials that trigger nerve impulses. Awareness of stimulus or sensation and interpretation or perception of the meaning of stimulus occurs in the brain. Then receptors can be classified based on type of stimulus, body location, structural complexity, and adaptation. Classification by stimulus type. Classification by location. Exteroceptors respond to stimuli arising outside the body. Receptors in skin for touch, pressure, pain, and temperature. Interoceptors, in the sense visceroceptors, visceral, respond to stimuli arising in internal visit and blood vessels, sensitive to changes, chemical changes, tissue stretch, and temperature changes. Then proprioceptors respond to stretch in skeletal muscles, tendons, joints, ligaments, and connective tissue coverings of bones and muscles inform brain of one's movement. And classification by structural complexity, in the sense complex receptors, special sensory organs, vision, hearing, olfaction, gustatory, and equilibrium. Simple receptors for general senses. General senses include tactile senses, temperature sensing, pain sensing. They are unencapsulated, free or encapsulated nerve endings. In classification by adaptation, slowly adapting includes Merkel and Ruffini corpuscles and organs of some free nerve endings. Then intermediate adapting includes some free nerve endings. Slowly adapting includes Merkel and Ruffini corpuscles and organs and some free nerve endings. Then intermediate adapting includes some free nerve endings. Then rapidly adapting includes Meissner's corpuscles and end organs, Pacinian corpuscles, end organs, hair follicle receptors, and some free nerve endings. Differential sensitivities. Each type of receptor is highly sensitive to one type of stimulus for what it is designed and yet is almost non-responsive to other types of sensory stimuli. The pros and cons of the eyes are highly responsive to light but are almost completely non-responsive to normal ranges of heat, cold, pressure on the eyeballs, or chemical changes in the blood. Mechanoreceptors. There are three classes of mechanoreceptors: tactile, proprioceptors, and baroreceptors. Tactile receptors, it's an end organ as a Meissner's corpuscle or a Pacinian corpuscle, then that responds to light touch. Then baroreceptors, a type of mechanoreceptors allowing for the relay of information derived from blood pressure within the autonomic nervous system, located in carotid sinus and in the aortic arch. Then proprioceptor, a sensory receptor found chiefly in muscles, tendons, joints, and the inner ear that detect the motion or position of the body or a limb by responding to stimuli arising within the organism. Mechanoreceptors, skin tactile sensibilities, epidermis and dermis, free nerve endings, expanded tip endings, Merkel discs, plus several other variants, spray endings, then Ruffini's endings, encapsulated endings, Meissner's capsules, then crosses corpuscles, hair and end organs. Deep tissue sensibilities, free nerve endings, expanded tip endings, spray endings, Ruffini's endings, encapsulated endings, Pacinian corpuscles, plus a few other variants, muscle endings, muscle spindles, Golgi tendon receptors, hearing sound receptors of cochlea, equilibrium, vestibular apparatus, vestibular receptors, arterial pressure, baroreceptors of carotid sinus and aorta. Merkel receptors, Pacinian corpuscles, then Meissner's corpuscles, epidermis, dermis, subcutaneous tissue, Ruffini cylinder. Then transduction of sensory stimuli into nerve impulses. Whatever the type of stimulus that excites the receptor, its immediate effect is to change the membrane electrical potential of the receptor. This change in potential is called a receptor potential. The basic cause of change in membrane potential is a change in membrane permeability of the receptor, which allows ions to diffuse more or less readily through the membrane and thereby to change the transmembrane potential. Mechanisms of receptor potential: by mechanical deformation of receptor, by application of chemical to the membrane, then by change of the temperature of the membrane, by the effects of electromagnetic radiation. Maximum receptor potential amplitude. The maximum amplitude of most sensory receptor potentials is about 100 millivolts, but this level occurs only at an extremely high intensity of sensory stimulus. Stimulus strength, amplitude of observed receptor potential percentage, slight increase will be there. Then receptor potential to action potentials. When the receptor potential rises above the threshold for eliciting action potentials in the nerve fibers attached to the receptor, then action potentials occur. More the receptor potential rises above the threshold level, the greater become the action potential frequency. Then receptor potential of the Pacinian corpuscles. The receptor potential include a local circuit of current flow. Receptor potential, deformed area. Then action potential, node of runway. Nerve fibers that transmit different types of signals and their physiological classification. Unmyelinated fiber, partly myelinated fiber B, individual axons, fully myelinated fiber C. Atypical non-myelinated and myelinated nerve fibers. Next, this picture is a very important one. Central nervous system connections. Free nerve endings, receptor, peripheral process, sensory ganglion, central process, central nervous system. Then encapsulated nerve endings, connective tissue capsule, glial cells, sensory cells, glial cells, then peripheral process, then central processes. Spatial and temporal summation. The different gradations of intensity can be transmitted either by using increasing number of parallel fibers or by sending more action potentials along a single fiber. These two mechanisms are called respectively spatial summation and temporal summation. Spatial summation in the sense, temporal summation here we could see. Spatial summation, simultaneous stimulation of many presynaptic terminals, action potential in initial segment of postsynaptic neuron. Temporal summation, repeated stimulation of one presynaptic terminus, action potential in initial segment of postsynaptic neuron. Detection and transmission of tactile sensations. Transmission of tactile signals in peripheral nerve fibers. Almost all specialized sensory receptors such as Meissner's corpuscles, logodome receptors, hair receptors, Pacinian corpuscles, and Ruffini's endings transmit their signals in type A beta nerve fibers that have transmission velocities ranging from 30 to 70 m per second. Conversely, free nerve endings, tactile receptors transmit signals mainly by way of a small type of A delta myelinated fibers that conduct at velocities of about 5 to 30 m per second. Some tactile free nerve endings transmit by way of type C and myelinated fibers at velocities from a fraction of a meter up to 2 meter per second. These nerve endings send signals into the spinal cord and lower brainstem, probably observing mainly the sensation of tickle. Then thus, the more critical types of sensory signals, those that help to determine precise localization on the skin, minute gradations of intensity or rapid changes in sensory signal intensity, are all transmitted in more rapidly conducting types of sensory nerve fibers. Conversely, the cruder types of signals such as pressure, poorly localized touch, and especially tickle are transmitted by way of such slower, very small nerve fibers that require much less space in the nerve bundle than the fast fibers. Pacinian corpuscles can detect signal variations from 30 to 800 cycles per second because they respond extremely rapidly to minute and rapid deformations of the tissues. They also transmit their signals over time. A beta nerve fibers, which can transmit as many as 1000 impulses per second. Low frequency vibrations from 2 to 80 cycles per second, in contrast, stimulate other tactile receptors, especially Meissner's corpuscles, which adapt less rapidly than do Pacinian corpuscles. Detection of tickle and itch. The sensations are transmitted by very small type C unmyelinated fibers similar to those that transmit the itching slow type of pain. Itch can be relieved by scratching if this action removes the irritant or if the scratch is strong enough to elicit pain. The pain signals are believed to suppress the itch signals in the code by lateral inhibition. Then sensory pathways from the entry point into the code, and then to the brain. The sensory signals are carried through one of the two alternative sensory pathways, the dorsal column medial lemniscal system or the anterolateral system. These two systems come back together partially at the level of the thalamus. Dorsal column medial lemniscal pathway. Anterolateral pathway. Dorsal column medial lemniscal system. Dorsal column medial lemniscal system. A Upon entering the spinal cord through the spinal nerve dorsal roots, the large myelinated fibers from the specialized mechanoreceptors divide almost immediately to form a medial branch and a lateral branch. The medial branch runs medially first and then upward in the dorsal column, proceeding by way of a dorsal column pathway all the way to the brain. The lateral branch enters the dorsal horn of the cold gray matter, which divides many times to provide terminals that synapse with local neurons in the intermediate and anterior portions of the cold gray matter. Then begins the crossing of the medial lemon cell in the medulla. The left side of the body is represented in the right side of the thalamus, and the right side of the body is represented in the left side of the thalamus. That is how it's arranged. Then somatosensory cortex. Human cerebral cortex is divided into about 50 distinct areas called Brodmann's areas. Sensory signals from all modalities of sensation terminate in the cerebral cortex immediately posterior to the cerebral central fissure. The anterior half of the parietal lobe is concerned almost entirely with perception and interpretation of somatosensory signals, but the posterior half of the parietal lobe provides still higher levels of interpretation. Visual signals terminate in the occipital lobe and auditory signals terminate in the temporal lobe. The position of the cerebral cortex anterior to the central fissure and constituting the posterior half of the frontal lobe is called the motor receptor and is devoted almost entirely to control the muscle contractions and body movements. Somatosensory areas 1 and 2. Two separate sensory areas in the anterior parietal lobe. The term somatosensory cortex almost always means area 1. Layers of somatosensory cortex. The cerebral cortex contains 6 layers of neurons, beginning the layer 1 next to the brain surface and extending progressively deeper to layer 4. The incoming sensory signals excites neuronal layer 4 first, and the signal then spreads towards the surface of the cortex and also toward deeper layers. Layers 1 and 2 diffuse non-specific input signals from lower brain centers that facilitate specific regions in the cortex. The neurons in layer 1 and 3 send axons to the related portions of the cerebral cortex in the opposite side of the brain through the corpus callosum. The neurons in layer 4 and 5 send axons 5 and 6 send axons to the deeper parts of the nerve system. Then functions of somatosensory area 1. Widespread bilateral excision of somatosensory area 1 causes loss of the following types of sensory judgment. The person is unable to localize discretely the different sensations in the different parts of the body. The person is unable to judge critical degrees of pressure against the body. The person is unable to judge the weights of objects. The person is unable to judge shapes or forms of objects. This condition is called asterognosis. The person is unable to judge texture and of materials because its type of judgment depends on highly critical sensations caused by movement of the fingers over the surface to be judged. Then anterolateral pathway transmits sensory signals that do not require highly discrete localization of the signal source and do not require discrimination of the fine gradations of intensity. These types of signals include pain, heat, cold, crude, tactile, tickle, itch, and sexual sensations. Characteristics of transmission. The velocities of transmission are only 1/3 to 1/2 in dorsal column medial lemon cell system, ranging between 8 and 40 m per second. The degree of spatial localization of signal is poor. The gradations of intensities are also far less accurate, with most of the sensations being recognized in 10 to 20 gradations of strength rather than many as 100 gradations of the dorsal column system. The ability to transmit rapidly changing or rapidly repetitive signals is poor. Then Pacinian corpuscles lies in the deep dermis and subcutaneous tissue. Characteristic lamellar and capsular wrappings. Perineural capsule is organized into 30 or more concentric layers of cells and fibrous connective tissue. The middle subcapsular zone is composed of collagen and fibroblasts. The inner core consists of Schwann cell mediated hemimembrane and they serve as rapidly adapting mechanoreceptors response briefly to beginning and end of stimuli, then detects pressure changes especially to vibrational stimuli. Here we could see a picture of Pacinian corpuscles. Acinous corpuscles highly concentrated in areas sensitive to light and touch. Fingertips, lips, tongue, soles, genitals, capsule and inner core located in the medial papilla. In the dermal papilla, they are oval or elongated mechanoreceptors. 1 to 6 axons enter the corpuscle, ramify extensively, and terminate in bulboid endings that are surrounded by lamellae. Listeners corpuscles. Merkel's disc. Slow adaptation because of rigid structure, sustainable response in humans 30 minutes, regular firing sustained, large receptor field, small sharp pressure, fast firing rate, large flat pressure, slow firing rate. Location in hair, less skin and hair follicles, note in skin surrounding the follicle. Free nerve endings are also associated with individual Merkel cells. Merkel cell nerve complex is also known as touchstones. Hedaciform endings, Lagos capsule, Pinbus corpuscles, SLB. I'm not sure about the pronunciation of all this. In the palmoplanar plantar skin, these complexes are found at the sites where the encrine sweat duct penetrates a glandular epidermal papilla. Merkel cells, tactile disc. Ruffini endings, only in the glabrous skin, sensitive to skin stretching, attributes to fine motor control, contributes significantly to finger positioning and movement.